let's start modeling meiosis now, and we'll use the same dihybrid that we just introduced with these different inputs forming it. Now, there's two possible options. These loci could be on separate chromosomes, which is what we'll model first, and then after that we'll model them as if they were linked on the same chromosome. Now, for an organism that's 2N, as any cell that can undergo meiosis has to be, for any cell that's 2N, we can draw chromosomes in pairs. And I'll switch colors for the second chromosome. I'm also going to deliberately draw these chromosomes as different sizes to indicate to me, or to remind me rather, that I'm dealing with two distinct chromosomes. So this is an organism that's 2N. If we wanted to make an organism that was N, it would just be one of the sets. So one copy of every chromosome is an N set, and taken together is a cell that is 2N. Now, in terms of loci, let's simply just put one locus, put the A locus on the one, and put the B locus, say, on the other. Now, this would have been, these chromosomes here would be the one input coming from the one parent, and this one here would be the second input coming from the other parent. So taken together, now we have an organism that's 2N and a dihybrid. The next thing you have to ask is, how do we model the steps of meiosis? Now, you might be familiar with the different cytological steps of meiosis from high school, where you've got, chromosome condensation and the different cytological changes that are going on. We're not at all interested in those in this course. We're strictly interested in what is going on genetically. The first thing that happens with a cell that's going to undergo meiosis is that the chromosomes are replicated, and they stay attached to one another at the centromere. That's what these circles are representing. That's a centromere. The centromere, just as a reminder, is the region of the chromosome that the cytoskeleton can grab onto for physically moving chromosomes to poles of the cell. So that's what that represents. We represent chromosomes with a single line, a circle for a centromere, and then any locus that we wish to put on, we put a hash mark across the chromosome. Now, maybe just to take a little bit of a digression here for a second, what would we see if we could actually, zoom in with a microscope and see what we're representing here? This is, if we sort of take this and blow it up, this is a double-stranded molecule. So it's a double helix with the two complementary strands with the monomers facing each other and base pairing based on hydrogen bonds. A centromere is simply a special sequence of DNA monomers which serves as a recognition site for cytoskeletal attachment. If we were to look at this and blow this up, from a molecular biology perspective, this is most likely perhaps, say, a protein coding gene. So, and again, this is a model that you're sort of familiar with from, say, cell biology, that sort of thing, where there's coding sequence, non-coding sequence that's transcribed, introns and exons that are part of the gene, so introns being spliced out and so on. And if I'm, say, representing the coding sequence with this dark filled in, the shaded in areas, so you can see that there's a start of transcription and then there's a start of translation, and it's going to make RNA and make a functional protein of some kind, that sort of thing. That's what we're representing in classical genetics shorthand with that little hash mark to say, here's the location of a gene that has some function that we're representing with this letter. Okay, so when a chromosome is replicated, as we're getting ready for meiosis, the very first thing that's happening is we've actually got the two copies. So each of these is a double-stranded molecule, and they're being held together physically with this protein complex at the centromere. All right, let's replicate our other chromosome. The next thing that happens is that these chromosomes line up with each other and find each other. And I've actually already drawn them that way, but the replication process takes place without their partners being next to each other. So this stage now is called synapsis, and in some cases we will represent synapsis by using a dashed line between the chromosomes. Synapsis is another protein-containing complex which is now physically associating the different homologous chromosomes with one another. We say that these chromosomes are homologous to each other because they have the same loci, the same basic sequence along their length with the different loci in the same order, although there may be slight variations in sequence, obviously, because we can have different alleles present. So homologous chromosomes find each other and pair with one another, and that's called synapsis, and that's our starting point for the next steps of meiosis, which is what we'll turn to next.